This is the lecture for Lab 11, Carbohydrate Metabolism. When presented with a sample of unknown bacteria, often the first step is to perform a streak plate. And the purpose of a streak plate is to isolate bacteria and to allow them to grow into a colony. So a colony is a mass of bacteria, identical bacteria. And from a streak plate, you can get colony morphology. And some of the important pieces of information from colony morphology is color, and also size and shape and texture. Often, after a streak plate has been performed, the next process is a gram stain. And a gram stain is going to identify what type of cell wall the bacteria has. And most bacteria are divided into either gram-positive bacteria or gram-negative bacteria. And in addition to identifying the type of cell wall, a gram stain also allows you to determine the cell shape. And with bacterial cells, for the vast majority of bacterial cells, there are three basic shapes: cocci, bacilli, and spiral. So doing these two tests, a streak plate and a gram stain, is very important because it's going to help you narrow down your options. So first of all, with a streak plate, most of the bacteria that you will be looking at produce colonies that are similar to this: small, circular, entire, and whitish colored colonies. For the gram stain, your bacteria are either going to be gram-positive or gram-negative. If you determine your bacteria is gram-positive, that means any type of bacteria that is a gram-negative is not an option for the identity of your unknown. Also shape, if you determine yours is a coccus, that means any type of bacteria that is a bacillus or a spiral is not a candidate for the identity. But once you have done a streak plate and a gram stain, this has just narrowed your options, but it will not give you a positive identity because there are hundreds of different types of gram-positive cocci that produce colonies similar to that. Once you have used the data from the streak plate and the gram stain to narrow down the options for your unknown, then the next step is to develop a biochemical profile. And biochemical profile is also sometimes referred to as an enzyme profile. And basically, this is a profile of all the different enzymes or all the different biochemical reactions that bacteria can perform. And once you have developed a biochemical profile, then you will compare the profile of your unknown to the profiles of known bacteria. And by doing this, you will be able to identify your unknown. Metabolism is defined as all the biochemical reactions that occur in a cell. And bacterial cells, like all cells, are constantly performing biochemical reactions, anabolic reactions and catabolic reactions. And for an enzyme profile or a biochemical profile, what we are trying to determine are what reactions a particular type of bacteria can perform. So we're trying to identify the different types of enzymes that are present in a bacteria and then compare that profile to known profiles. Now, of course, we are unable to actually see if these enzymes are present, so instead we need to use indicators. So some sort of indicator that this reaction is occurring. The focus of this lab is on carbohydrate metabolism, and carbohydrate metabolism involves taking different types of carbohydrates, running them through a reaction to produce ATP. And ATP is the readily usable form of energy for a cell. And all cells need a constant supply of ATP. And when carbohydrate metabolism is performed, often the waste products of ATP production are acidic. So often when cells are metabolizing carbohydrates, they will produce acidic waste products. And that's different compared to protein metabolism. If cells are forced to use proteins as a source of energy, often those waste products are alkaline. So for this lab, you are going to be investigating if your unknown bacteria has the ability to break down different types of carbohydrates to produce the ATP that the cell needs. And the indicator you are going to use in most cases are pH indicators, which will indicate if there's a decrease in the pH during the reaction. The first type of indicator you will use in this lab is Phenol Red. And Phenol Red is a pH indicator that is red at a pH of 7.4. And as the pH decreases and the solution becomes more acidic, it turns yellow. As the pH increases, the indicator becomes more pink. And in lab, you are going to be using Phenol Red for 4 different types of sugars: glucose, lactose, sucrose, and mannitol. If the bacteria are able to utilize the sugar to produce ATP, they will also be producing acidic waste products. So over the time of the incubation, the media will turn yellow. And that is an indicator that your bacteria can utilize that sugar. Also, in the glucose tube, you will have a small test tube inverted in the glucose test tube, and that is called the Durham tube. And the purpose of the Durham tube is to collect gas that may be produced if the bacteria are fermenting the glucose. So remember, with alcohol fermentation, you get gas production, and lactic acid fermentation, there's no gas production. So if gas is produced, you will see a clearing in the Durham tube. So this is a picture of a tube, glucose tube, that is positive for glucose fermentation and positive for gas production. This tube is also positive for glucose utilization, but it does not have a bubble, so it is negative for gas. And this tube is negative for glucose fermentation because the media still remains a red color. Acidic byproducts were not produced. Another test you will be performing is starch hydrolysis. Hydrolysis, this is a catabolic reaction. Basically, you are testing to see if the bacteria has the ability to break down starch, which is a large polysaccharide made of glucose molecules. The indicator you will be using for this is iodine. So iodine is normally sort of a brown color, but when it interacts with starch, it turns black. And for this lab, you will inoculate the bacteria onto starch agar. So the agar contains starch. So you'll inoculate the bacteria in a straight line. And after the bacteria have incubated for 48 hours, then you will add starch on top of the agar. And because the agar contains starch, the iodine should bind to the starch and create a black color. If your bacteria have the ability to secrete an enzyme to break down starch, then you should have depleted the starch around where the bacteria have grown. So when you add the iodine, you will have a light brownish color around the normal iodine color, which indicates that the starch around the bacteria has been broken down and it is no longer there. The other test is the Methyl Red-Voges test, and for this the indicator is methyl red. And methyl red has different colors compared to phenol red. When solutions are acidic, the color will be red, and when solutions are basic, the color of the solution will be yellow. And the Methyl Red-Voges test is testing how glucose can be metabolized. Often when glucose is metabolized, it will produce acidic waste products. But some bacteria have the enzymes to convert those acidic waste products into neutral waste products. So this is a test to determine what type of waste products are produced when bacteria metabolize glucose. Are they acidic or are they neutral? And this is an example of a methyl red test. Methyl red test, yellow is negative and red is a positive result. And this is an example of a VP test. So you're going to run 2 tests off this test tube. And the yellow, again, is negative and the red is positive. And VP tests are a little bit difficult to identify because when you add those reagents, you're going to get black precipitates. So you really have to look more at just the color of the solution to determine if the reaction is positive or negative. The last test you will be performing is the citrate utilization test. And this test is a little bit different than the other tests. First of all, the pH indicator you will be using is bromthiamyl blue. And bromthiamyl blue is a green color at a neutral pH. It will turn yellow at an acidic pH, and it will turn blue at a basic pH. And essentially, in the citrate utilization test, you are testing to see if the bacteria are capable of using citrate as a source of carbon if carbohydrates are not present. So most cells, bacteria included, prefer using carbohydrates to produce ATP. But if carbohydrates are not present, then you are forcing the bacterial cells to use other types of macromolecules to produce energy. And in this case, you are trying to determine if your bacterium can use citrate as a source of carbon to produce ATP. And also in the citrate agar, there are ammonium salts, which the bacterium would also have to be able to utilize in order to use citrate as a source of carbon. So if the bacteria do have the enzymes to use the ammonium salts and the citrate, then the media will turn basic because ammonia will be produced. So for the results, the original color of the media is a green, it's a neutral, and if it remains green, that means your bacteria are negative for being able to use citrate. If instead the media turns blue, that means the pH has increased because the bacteria produced ammonia as a waste product, and that indicates a positive.